Um, so if you were to take a look back here, remember we started to talk about constitutive, inducible, repressible, how bacteria are making sure that the genes that they need are on, and how they're making sure that the genes that they need at a specific time are ready only at the time that they need them. Because remember, making genes when you don't need them is a waste of resources and energy, right? So these are important regulatory mechanisms within cells. Put this up here. We talked about this already. All right, so this is where we ended, right? Repressors and activators. So repressors, right, are going to block transcription, right? So when we're blocking transcription, what enzyme are we blocking? Noel?
The RNA or not synthase, polymerase or something like that.
RNA polymerase, right?
Oh,
RNA polymerase is interacting with DNA to make messenger RNA. All right, so if we're blocking transcription, we're blocking an RNA polymerase from being able to bind to DNA. All right, so and you'll see this all kind of makes sense in case you're having trouble visualizing. But a repressor is going to bind to an operator, right? So the operator is downstream of the promoter. All right, so the promoter, right, is where RNA polymerase that Noel just mentioned binds. So the operator interferes, right? It almost kind of sets a roadblock, if you will, to the promoter. You then also have activators, right? In the case of activators, all right, there is an activation site, right, or an activator binding site that is going to help RNA polymerase bind to the promoter. So in this case, there might not be anything blocking. Hey, Emma McKenzie, thank you for my tea. I appreciate that. I'll see you later. Mwah. All right. So in this case, nothing's maybe blocking RNA polymerase, but it can't do it on its own. And so kind of a similar idea to a sigma factor, except this isn't a sigma factor. Okay. So here, right, is a little bit of an animation of what's going on. Okay. So here you have, right, a repressor, right? It's bound downstream, so it's kind of setting up a roadblock, all right, for that RNA polymerase. So this is RNA polymerase. I originally got this slide from Dr. Chrisman. So those of you that are nerds and like Voltron, which I never actually watched, but I've kept it because I think it's kind of fun. Right, this is my RNA polymerase. So RNA polymerase is bound to the promoter. We have a repressor though that's around the operator. And notice down here, this would be the terminator sequence, right? So when that RNA polymerase tries to move down, it hits the repressor and it's knocked off, right? It can't pass. Now, how do we get this repressor off? We actually then need something called an inducer. And an inducer is going to bind to an allosteric site. Those of you that are enzymatic experts, those of you that paid attention in ANP1 with me, let's see, Noel, Sarah, way back in the day. I think those are the only other people I had in AP. Caroline, did I have you? I had you in AP1, didn't I? Good, so you should know this. Mercy, I think. The allosteric site, if you guys remember non-competitive enzymatic inhibition, that would be when a molecule binds away from the active site and causes a change in the shape of that active site so that the substrate can no longer bind in, right? So you hopefully remember that. Sarah, do you remember that? You don't? You know who would? Kenneth. All right. So in this case, right, an inducer comes in and it binds to that repressor, right? It causes a change in the shape of that repressor, and the repressor comes off, and then transcription can occur. Okay, and we'll talk about some different examples here or specific examples in reference to that lac operon, as to what the inducer is, right? The inducer, there's not necessarily a universal inducer for these different genes, right? The inducer is going to be a lot of times specific to something related to what that gene product is going to be affecting. And you'll see what that looks like in just a second. All right. So in some cases, so right now we see transcription going on, right? It's constitutive. It just keeps on going and going and going. We want to turn it off, right? So how do we turn it off? Well, we need a repressor again, but in this case, we need something to help the repressor, right, get to the operator. So we have a co-repressor, right? I'm sorry, there's not a lot of difference here. It kind of looks exactly like my inducer from the last thing. Sometimes my level of creativity is average to below average, right? So just deal with it and make sure you're paying attention to the slide, right? So this co-repressor binds to the repressor. It allows it to attach to the operator, and then transcription is blocked. All right. Now, this is from your book. I'm sorry I don't have a little fancy art thing for this. Maybe I'll try to get that ready for microbiology in the fall, right? But this is the activator, and this is actually looking at kind of a similar situation that we'll talk about for the lac operon in just a second. But here you see the activator site is upstream. You see the promoter here, and in this case, the activator is not bound, so the promoter cannot bind. Now, to try to make this more confusing for you guys, all right, in order for the activator to bind, you have to have an inducer, all right? So the inducer binds to the activator, and the activator is then able to bind upstream. All right? So you have a couple inducers. You have an inducer that knocks off, right? You have an inducing molecule that knocks off a repressor. You can also have an inducing molecule that binds to the activator, activates it, so it can bind to the activation site, and then your RNA polymerase is able to bind to the promoter and start transcription of that gene. All right. So I actually think this is relatively straightforward if you kind of take the time to even draw out or write out what's going on kind of in your different situations. If this is the last time you look at it, I guarantee you're going to start to kind of mix these things together and glob them together. And I'll try to kind of give you some examples of these in the lac operon. The one thing I don't like, and I haven't really looked too hard in the new textbook that you guys are using, so I might go check out this specific section. But in the old textbook that I had you guys use, what it didn't do a great job of doing, and you'll see this in the lac operon in just a second, is both of these things, or these things don't necessarily have to happen independently. So you'll see in the lac operon, you're going to have an activator that binds for the polymerase to bind. You're also going to have a repressor removed in order for that polymerase to work, right? So these are not necessarily independent events. You can need an activator and have to have a repressor removed. And so there can be a lot of things going on. And that's important too, because that's multiple touch points and multiple regulation points for cells to use. Okay? All right. So this is the lac operon. What it's not showing here, and we'll talk about this in just a second, is that in order for this polymerase to bind, there actually was an activator that also had to bind, all right? And that would actually be in this situation down here, because we'll talk about that activator specifically. So, ooh, look at this. Enzyme, 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 all important for the utilization of lactose as an energy source. Um, Glory, do you remember the term when we have genes stacked up back to back to back and we can transcribe them all at once?
Um, I can't remember.
You remember the term for many that we talked about in NEP1 when we talked about like if you have a like a complex sugar? Do you remember what its other name is?
No, I'm sorry.
That's okay, Glory. I can't stay mad at you for too long. Can someone help her out? Does anyone remember what this term would be for these genes? Or if you can't remember what the term for the genes is, what a complex sugar is also called?
Could be like poly.
Okay, so polys for so like a polysaccharide, right, is a complex sugar. So that's the first part of this answer. So poly what for these multiple genes?
Oh, I'm so sorry. I just interrupted you. Is it polycistronic?
Polycistronic, right? So genes back to back to back. So this is incredibly efficient, right? All of these enzymes are needed for lactose utilization. So bing, bang, boom, one transcript, and you are ready to go. Now, in this case, right, and you'll see this in a little bit more detail here in just a second, a repressor is bound, right? And we'll talk about why that is in a minute, but a repressor is bound. And so RNA polymerase cannot get to these gene products. All right. And you can see here, there's no lactose in the cell. So if there's no lactose in the cell, why would you want to be wasting time and energy making the enzymes that are needed? Whew, that's hot. All right. In this case, though, it gives you this alternative situation. Lactose is now present in the cell. Right? So in this case, it's telling you glucose is not available. That could just be that it's not present, or we'll see a scenario here in just a second. Glucose was present, but you've used it all up. Okay. And once glucose is gone, and if lactose is present, when lactose enters the cell, some lactose, right, kind of spontaneously converts into a molecule called allolactose, right? It's a variant of lactose. And what do you know? Allolactose happens to be the inducer that's needed to knock off this repressor. So again, I kind of mentioned last slide, there's not just this magical inducer that's floating around and knocking off repressors. The inducer is going to have some type of relationship, right, to the genes that you're transcribing, right? So these genes that you're going to transcribe are for lactose. And so allolactose is a derivative of lactose. So it's the inducer that knocks off this repressor. Once the repressor is removed, RNA polymerase can do its job, and you now have your enzymes present that are going to let you utilize lactose as an energy source. All right? That makes sense? Yes? Okay. It's about to get a little more, I'm going to kind of build off of this. All right. So please, at some point during this, if you get a little lost or confused, let me know. Now, this should be familiar to you and hopefully should not scare you very much. Caroline, are you scared?
A little bit. That's okay.
Okay, good. A little bit. A little fear is good, right? Keeps you on your toes and ready to learn. So what you're seeing, right, is essentially a double growth curve. And what we had before, right? Here's our lag phase. Here's our log phase. And what you guys noticed before, right? And you guys all did relatively well on this question. Some of you, the only thing that I saw a couple times is some of you left off the prolonged decline phase was kind of the one major thing. And a couple people were saying that bacteria was growing during the lag phase. which if this is the number of cells, right? So this represents fewer and more. If it's flat, there's no growth. And it's important to point out here, well, actually, Lauren, why don't you do it for me? Lauren, what happens in the lag phase again? What's going on right there?
Enzymes are synthesized.
Right, enzymes are being synthesized so that you can utilize the nutrient sources present in your environment, right? So while you're kind of gearing up, there's no growth that's occurring. So what we saw before is lag phase, log phase. You guys then had a stationary phase. You had a little bit of an exponential decline for the death phase, and then kind of a prolonged decline that kind of tailed off. But now we see, right, that in the presence of two nutrients, things change. So if we wanted to go back and look at our original growth curve, it was kind of assuming that there's only one nutrient present in the environment. And that might not be true in nature, but it could be true in a laboratory setting, right? You might only have one type of sugar in there. And so when it uses it up, it's gone. But in this case, we're actually saying that, hey, there are two energy sources present. So here's glucose and lactose added at the same time. So you gear up for enzymatic acquisition or utilization of those nutrients. So there's no growth. You then have an exponential growth, right? Glucose, right? This is an important thing to point out is glucose for most cells is the preferred energy source, right? The enzymes that are constitutively expressed and turned on are those that are utilizing glucose, right? And so we get enough of those enzymes ready to go. We utilize glucose, exponential growth. We then go into what would be the stationary phase, but the cell is able to detect and realize that lactose is present. And so we have another lag phase. So during this lag phase, right, that's when we're making enzymes. Just like on the previous slide, we are making those enzymes that are needed for utilization of lactose, right? We get them, they're good to go. We have exponential growth using lactose as an energy source. Lactose is used up, right? This would then be a stationary phase. We'd enter that death phase, right? That exponential decline that's relatively brief. And then we'd go into our prolonged decline. Elena, can you remind me what happens in a prolonged decline?
Don't the cells become, oh goodness, endospores, or they can create endospores in that time, right? Or
So that's not exactly what I was looking for. But during death and prolonged decline, if you are a bacillus or a clostridium, you might be able to do that. Yes, that's true. So that's not exactly what I was looking for, but that's not wrong either. Who can finish it off for Elena though? What exactly was I looking for in relationship to the test? Oh, wait.
So
Oh, nope. This is Anna's. She's ready.
Okay.
Anna, what happens in prolonged decline?
Aren't they kind of like shifting into different types of like variations of the bacteria?
Right. There's adaptations, right? They are potentially changing their metabolic pathways. There might be mutations that are occurring. If you remember, we talked about this during the growth curve. This would be one of the worst times for you to test your bacteria because metabolically, they don't resemble the original bacteria, and your tests are going to kind of be thrown off. You might get results that are misleading you or kind of head scratchers. And so prolonged decline, we see adaptation and changes metabolically that make it more challenging to identify those bacteria. All right, so the double growth curve. So I want you to know this, right? And you guys should, hopefully, if you don't, please maybe look over this tonight during some of your studies. I've emphasized this over and over and over again. That means it will come back to you, right, is what, right, is happening during both of these phases of exponential growth, right? What happens within a bacteria that allows that to happen, right? You guys should know replication, transcription, translation, mRNA processing, right? What's going on in prokaryotic cells that allows them to grow this rapidly in a way that we can't do, all right? So in relationship to the lac operon, one of the things that you hopefully were thinking of, even though I mentioned, hey, glucose is the preferred energy sources, what ultimately, right, what ultimately is preventing multiple energy sources from being utilized at once, right? And so that's where you see carbon catabolite repression, CCR. Where else have you guys heard of CCR? No one? No one? Professor Perry, where have you heard of CCR? All right, you're muted.
Sorry, I couldn't get unmuted. I'm not sure what you're talking about. Besides this.
I'll give you a clue. John Fogerty?
What?
John Fogerty? Oh, Professor Perry, you let me down. I'm going to tell Dr. Chrisman about this, right? CCR.
I'm sorry.
Creedence Clearwater Revival.
Oh,
Make a joke, Dr. Daft.
Yeah, that's a good one. Come on.
That's even, well, I want to say it's before my time, but not really.
If you haven't heard of them, though, they're a really good band. A lot of their music is still used in movies. And any of you that assuming, again, this is none of you, but I could be wrong, right? I hope that I'm wrong. Any of you that are interested in Vietnam history, their band, they were kind of big in the Vietnam era and even a little bit after that. So if you ever watch any Vietnam movies, there's usually CCR playing in the background, right? Please don't include that in your answers on the exam. Well, I guess I don't mind if it's in there as long as it's at the end and everything else before it's right. Deal? But carbon catabolite repression is basically referring to, and I have it written right here, one energy source at a time, right? So it is what is allowing, right, a cell to focus on one organic carbon or one organic energy source at a time. Because again, it's not energy efficient to be going after multiple energy sources if you have an abundance of one, even in the presence of another. And so this is how, right, in the presence of, right, and again, this goes into play for other sugars, but we're focusing on glucose and lactose. And so I'm kind of walk you through this. I think it makes sense if you take time to kind of move through it or try to move through it sequentially. Again, this is one of those things where after you sit down and look through it a couple times, it clicks. If you glance over it once in your notes or read through it once as you're studying, you might have some issues, because I guarantee you there will be a question about CCR, not the band, on your exam. So in this case, these little triangles are glucose, right? And so you are taking glucose into the cell, right? And you remember, right, I told you before when we were talking about the central metabolic pathways, one of the things that happens to glucose, because it helps you hold on to glucose, whether you're a prokaryotic cell or eukaryotic cell, is you phosphorylate glucose as it enters the cell, all right? And here doing that is something called a transporter component, all right? Now we could get more specific, and I think Dr. Chrisman does in his class with specific enzymatic names for these. I am looking for an understanding more of the overall process. And so calling a transporter component for me is totally fine. And what you see here is this transporter component has a phosphate group on it. As glucose enters, it donates its phosphate group to glucose, right? So you get glucose 6-phosphate. This unphosphorylated transporter component, right? I like to think of it as being lonely. And so it wants to bind to something. And what does it bind to? It binds to the lactose transporter, or also called lactose permease, all right? This is how lactose enters the cell. This unphosphorylated transporter component is blocking that channel. It's not allowing lactose to move in. So as long as glucose is present, that means that this transporter component is giving up its phosphate to glucose, and it's going to be blocking, right, this lactose transporter, this lactose channel. All right. Now, inevitably, right, at some point, as glucose starts to run out, this transporter component doesn't have anybody to give its phosphate to. So what does it do, right? This phosphorylated transporter component then goes and interacts with ATP, right? And turns ATP into something called cyclic AMP. If you want to know why it's cyclic, Google it, and you'll see that its structure is kind of circular, right? If you're expecting a perfect little circle, you're going to be disappointed, right? But it's a circle-like structure, so we call it cyclic AMP. Cyclic AMP becomes the inducer that is needed for the CAP activator. All right. So once cyclic AMP is made by that phosphorylated transporter component, because, right, it has its phosphate, because glucose is no longer coming in, all right, then we start to get, right, synthesis of these genes or transcription of these genes. All right. So a couple of things that I kind of want you to note and try to draw together here. Now that this transporter component is phosphorylated, it's no longer able to block this lactose transporter. So that means that as you're converting ATP into cyclic AMP, lactose is starting to come into the cell, right? And so now you are starting to make the enzymes that are needed to utilize lactose as an energy source. So here, right, this point right here, we're entering another lag phase as we kind of get enough of these enzymes available to properly utilize the lactose that's present. All right. And if you go back a few slides, what it's not showing here, and I always say this, and so maybe I'll actually do it this time. All right. Gloria, you hold me to it. All right. In the fall, when you see me, say, Professor Daft, did you adjust the CCR slide? All right. Promise? All right, cool. We'll see. I'm not going to forget. I'll write it in my notebook after this. But what they're not showing is, right, so we have this activator that's bound because of cyclic AMP, right, which is an inducer, binds the activator, activator binds the activation site, RNA polymerase can bind. So what's missing from this picture that you guys should be able to tie in from a couple slides ago? Megan, help me out. What's missing from this picture that they're not showing? And I don't know why they decided to not show it.
The repressor.
The repressor, right? And you remember what the inducer is for the repressor? I know this was just briefly, so it's kind of putting you on the spot a little bit. But what's the inducer that removes the repressor for the lac operon?
Allolactose.
Allolactose, perfect, right? So phosphorylated transporter component, no longer blocking the lactose transporter. Lactose starts coming in. Some of that lactose is converted to allolactose, right? Which binds to the repressor. It comes off. That phosphorylated transporter component converts ATP to cyclic AMP, which is an inducer for the activator. The activator binds, all right? So you have the activator bound, you have the repressor removed. RNA polymerase can now go down and transcribe the genes that are needed to utilize the energy source that is now flowing into the cell because glucose is no longer present. Now, again, glucose is the preferred energy source. And so if we were to dump glucose back into the system, we would start to have unphosphorylated transporter components. They would start to block this. You would turn off all of the transcription of lactose genes, and you start utilizing glucose again. All right, so glucose is that preferred energy source, but this whole mechanism makes sure, right, that you're only using one carbon source, one organic source of energy at a time, right? So it's efficient and it saves the cell energy. Does that make sense? Or will it make sense after you look over it. And I am serious. Please look over it. And if you have questions, if you're having trouble understanding, let me know, right? Because this kind of ties in some of these basic concepts of gene expression and gene repression. All right? Elena, what are you drinking there?
It's just coffee.
Yeah, I don't like coffee. I'm drinking green tea. My father-in-law got it on to me. I'm going to make it to 102.
Okay. I like green tea at night, but in the morning, I just need something.
I got you. My wife, she likes her some coffee too. So I feel I just never got into it, you know? And I'm not sad because coffee is an expensive habit if you like good coffee, right? Yeah, I know, right? It's just my mom, and maybe that's why I don't like coffee. I grew up, my mom would use like the Folgers crystals, you know, that you get in the little plastic thing and just dump those. And so she's not what I'd call a sophisticated coffee drinker. And so maybe I never got a taste of the good stuff. And here I am. All right, cool. So important concept. This is everything that we just talked about. I actually don't really spend much time. You can read about it in your textbook. MicroRNA or RNA interference. This is kind of a cool mechanism that can help regulate gene expression by actually degrading RNA. It's not that I don't think that it's important. I just don't think your textbook spends a lot of time on it. We don't really revisit it too much. And so if you would want to know more about that, and this is primarily a eukaryotic mechanism, by all means read on it, but I'm not going to really test you on it because your textbook kind of glosses over it. If you're more interested in that, then at some point, depending on your major, I would recommend taking like Cell Bio or Biochem 1 would go over these in a little bit more detail, right? So if you want to read about it, it is interesting. I just, you know, there's only so much time for so many things. And so I don't spend a lot of my time there. All right. So let me okay, cool. All right, so let's start to talk about mutations. And so this is kind of building off some of the basic foundational genetic information that you got in the last chapter. Again, I love opportunities for shameless pitches, right? If there's something that you're really struggling with or don't understand, please reach out to me and we can figure out a way to talk about that or discuss that with one another, right? But we're going to move into mutations. I think this is actually a pretty interesting chapter. And maybe I should retitle it kind of like your one activity, Mutations in the Sharing of Genetic Information, because we will talk about horizontal gene transfer in here, but I kind of group it in here because a lot of times the information that they're sharing that we really care about is some type of mutation that they've picked up along the way. So, you know, we'll talk a little bit about genetic stability and instability, kind of some unique features of prokaryotic cells that allow them to work and replicate very quickly. And truth be told, you'll see this in just a second. And this happens in us too. When replication is occurring, one transcription is occurring, one translation is occurring, mistakes can be made, all right? Even in us, it's moving at a relatively quick pace. And so if you guys have typed up, right, your papers, so your papers that I'm a little over halfway through and actually have been relatively enjoyable. I enjoy personal reflections and just kind of hear where you guys are coming from. But let's say you wrote that in like an hour, right? And you just write. Even though, right, you know how to spell, right? You make mistakes, right? Even when you guys proofread your papers, right, there are still mistakes that are made, right? There's a lot of words on that page that you've written and you're trying to proofread. And with the exception of maybe very few of you, right, there's always a mistake or two that you don't catch. And that's just life. And the same thing occurs, right, when we're processing some types of genetic information, right? They're really good at what they do, but they make mistakes. Just like we do when we're proofreading or editing or writing papers. Okay? So we'll talk about how mutations are occurring. We'll actually talk about some kind of unique repair mechanisms that bacteria have that allow them to survive situations we can't survive. And I would say appreciate bacterial genetics in the lab, but, right, there's a pandemic, so that's going to be a little challenging. But I'll try to, at least get you guys as much of the base knowledge as I can get. Again, I don't know what all of your background is. Those of you that have taken Bio 1 and Bio 2 have at least got a little bit of a taste of this. If you've had genetics already, then you've had this. A&P 2, I think this is one of the last units that you do. And I'll be honest, when I've taught A&P 2, this is usually the one that I rush the most or barely cover. So just a little bit of a quick review, terminology-wise, just so we're on the same page, right? So when we talk about a genotype, we're actually talking about the genetic sequence, right? What's the nucleotide sequence? And so when we talk about a change in genotype, right, here you can see that these are identical nucleotides, or identical nucleotide sequences, sorry. And here you can see that we've changed, right, a T and a C right here, right? A thymine and a cytosine, boom, boom. So we've changed those out. So that's a change in genetic sequence, right? Or a change in genotype, right? Now, a phenotype is a physical characteristic, right? So some basic phenotypes are alive versus dead, or color, or size. Some of these could be color blindness, all right, versus regular vision. Those of you that are maybe seniors, which I think there are some that maybe had, or are going to be seniors that had Bio 1 a few years ago. If you had it before, Dr. McCrae took over one of the things that we used to do when Dr. Riley was in charge. And if you don't know who that is, then that's fine. But we would do this little test where you'd have these little pieces of paper that were edible, and they would have different chemicals on them. And some people could taste it and some people couldn't, right? And so that is actually a phenotype. And so, sometimes I think the term physical characteristic can be a little misleading. I actually like a phenotype is something that you can kind of easily measure, right? You can measure somebody's ability to taste or not taste something. You can even measure somebody's ability to hear or not hear something, right? So there's a lot of different examples of phenotype. And one of the things that you're going to see here in just a second is a change in genotype does not necessarily have to be a change in phenotype. All right, so kind of keep that in mind as we start to move forward here. We'll get a few slides in and then we'll take a short little break. So when we talk about a mutation, we're comparing that back to a wild type. Now, as I went on doing research in grad school, and I'm sure that Professor Perry would agree with me too, based on some of the things that she's seen. Wild type, another way that we kind of look at it is, what's normal, right? And to me, normal is a relative kind of term, all right? And so sometimes when you hear me refer to normal, I'm referring to the wild type, even though that's not the most accurate description. But a lot of times, referring back to, or sometimes to normals or wild types considered like the healthy version. And so, why can't I think of it? Who presented today? Yeah. Oh, sorry, Rachel. I was like, who presented that microbiome article just a few minutes ago? So, Rachel was talking about the microbiome. And so one of the things that we look at in microbiome research is, so I looked at the microbiome as related to type 1 diabetes in grad school in my research. And we would compare a diabetic microbiome to a non-diabetic microbiome. So we consider the non-diabetic microbiome the wild type microbiome. But that doesn't necessarily mean that just because they're non-diabetic that they have a healthy microbiome, all right? And so to me, it's a relative term, but we just got to go with it. So when it comes to mutations, you have a couple different types. You have spontaneous, all right? So for those of you that like the X-Men, which I assume is all of you, right? Right, Lauren? Okay, good. Or at least you're good at humoring me, right? So the X-Men, if any of you ever were into that when you were younger or into it now, right, we would consider that most of these are spontaneous mutations. There's always expectation, or there's always exceptions. Some of them may have been induced, right? But spontaneous. It kind of happened without any intervention. So some mistakes were made while their DNA was being replicated or while their genes were being transcribed, and they ended up with these superpowers, all right? You then have induced, right, which is my favorite. So those of you that follow Ninja Turtle lore, right, a bunch of turtles are abandoned or dumped in a sewer when they're babies, and then a research company dumps some of its research product down into the sewer. The turtles interact with it. The turtles are also hanging out with a rat, and so that rat turns into Splinter, and the turtles turn into crime-fighting heroes, right? But there was some type of interaction with the environment that induced the mutations that they had, okay? And those of you who were paying attention, yes, I did have my Ninja Turtle shirt on Monday. Thank you for noticing. So really briefly, we'll talk about replication, and to just kind of give you an idea, maybe another feather in your cap for how bacteria are able to replicate so much quicker than we are. As you see here, for E. coli, it's just an example bacteria. They can replicate 1,000 base pairs per second. All right, that's fast. In eukaryotic cells, we do about 40 base pairs per second. All right. And it turns out that the error rate in prokaryotic cells is less than the error rate in our cells. So we make more mistakes, but because they replicate so much more quickly, right, they have a higher number of individual mistakes, right? So the way to look at this is that if that's kind of confusing, is we can use COVID cases as an example, all right? And so you look at the number of COVID cases, and I haven't checked recently what the percentages are, if they've shifted drastically, but Italy was running at about 10% death rate with COVID-19, between 10 and 11%. So I'm hoping or assuming that's come down a little bit since I last checked. And America was running somewhere between probably 4 and 6, all right? But we have a larger population. And so even though we had a lower death rate as far as a percentage, we had a higher number of deaths than Italy. Does that make sense? Right, because 4 to 6%, and we'll just do total population size of the countries, right? 4 to 6% of some place that's 300 million compared to, 10% of some place that's 6 million. Right, I'm just kind of spitballing pop, right? So that makes sense. So even though they're better, right, they make less errors because they are replicating so quickly and going through so many generations. There's just going to be more errors that are typically present, right? And so if you think about it, they're doing this all in minutes, you're doing this all in hours. And so they're just going to accumulate a higher total number of mistakes, even though the percentage of their mistakes is lower. All right, so just kind of let that sink in. It doesn't make sense. Let me know, and we can walk through that again. Now, what's really cool in prokaryotic cells is that spontaneous mutations, sometimes it's possible for reversion to occur, right? So this would be in us, like let's say you were born with cystic fibrosis, right? Because that's kind of a genetic disorder that we talk about a lot at Lee, right? Because we have the Race for the Roses, which I think is supposed to happen in the fall now, hopefully, if things continue to proceed forward in a good way. But that'd be like if someone's born with cystic fibrosis and then, I don't know, they're 10 years old and they convert back to the wild type, right? Their body changes, it gets the gene sequences right, and they're totally back to normal, right? That doesn't happen. We don't see that, but prokaryotic cells can do that. So they go back to their wild type. I assume since you didn't know CCR, you probably won't know who this is. He passed away a few years ago. This is David Bowie. Right, he's a pretty talented musician. He also appeared in a couple movies. If any of you have seen Labyrinth, which is done by Jim Henson, who made the Muppets, right? He's actually like the main villain in this, but this is David Bowie early in his career. This is David Bowie in the middle of his career. I just had Cocoa Puffs, and you know what Cocoa Puffs do to you, right? They make you cuckoo a little. And then this is David Bowie, right, later in his life. And so this is just a fun example of a reversion, right? This is what he started off as. There's a mutation, even though not really, right? And then he reverts back, to his old self, right? So it can go back to the wild type. That's not something that we see in us. So that can occur, but not going to really happen in eukaryotic cells. I don't know. Professor Perry, can fungi, can they revert? Can they mutate and revert back to a wild type? Do you know?
I do not.
Okay. That's a look.
I've not studied much about fungal genetics.
Not yet, right?
Right.
I don't know. I'd have to look. I would almost feel safe betting that if there's a eukaryotic cell that could do it, it might be a fungal cell, but I'd have to look at that. So right now we're just going with the assumption that this is something, though, that's primarily prokaryotic. Now, what we're going to talk about here is not primarily prokaryotic in the sense of these mutations that we're about to go over can also occur in eukaryotic cells. So this is where we're going to take our break. It's 9:06, so 9:15-ish. All right, and by ish, that means there's a 1 to 2 minute grace period there. 9:15-ish does not mean 9:30, Megan. So why don't we take a little bit of a break? We'll come back and we'll finish up here. I'm going to leave this on so you guys can stay on if you want, and just turn everything off. And I'm going to recharge this computer a little bit. So let's resume recording. All right. So let's start talking about different types of spontaneous mutations. When we talk about spontaneous mutations, there's a couple different options. One of the big ones being a base substitution. And this can be, right, as simple as one base, right? And when we substitute one base, we call this a point mutation, right? Mutation is occurring at that point where we've changed the base. And this generally keeps frame. If I would say something generally keeps frame, Allie, what am I referring to that we talked about yesterday?
The codon sequence.
Perfect, right? The codons that are being read, right? So every 3 nucleotides is a codon. If you're simply changing out one nucleotide for the other, then you're not affecting, right, that sequence of 3 nucleotides, which equals a codon. Okay? So you have 3 outcomes, and you'll see this as the case in all types of mutations: silent, missense, and nonsense. So for a silent mutation, this is a change in genotype that has no effect on the phenotype. And the reason this can occur is because you see this codon ACA all right? Same amino acid as ACG. What's that word that I used, Amanda, that referred to the fact that a lot of codons can code, or multiple codons can code for the same amino acid?
Degenerative?
Yes, degenerate, right? So degenerate, right? Multiple codons for the same amino acid. And this is actually really important because, right, every time we have a mutation, it doesn't necessarily have a negative effect. So by looking at you, by looking at me, right, these are mutations that we can't detect based on phenotype, right? We'd have to get down at a molecular level and analyze, right, your genetic sequence to figure out if you have these or not. So these are mutations that we don't really worry about, all right? And I think one of your questions in your textbook, right, referred to the fact that a large percentage of mutations are actually silent mutations. We just never know that they've occurred. And it's the mutations that have these really big kind of consequences or effects are actually the minority of mutations, all right? So missense, a possible effect on phenotype, right? So now you have two different amino acids. Sometimes, right, this protein that results after this mutation is going to have partial function. We also call this leaky, all right? So it may not be as efficient. You may have maybe half of your normal function. You could possibly have all of your normal function. We didn't spend time on it, but those of you that have gone over amino acids, at least briefly before, know that some of them, although they're different, share certain characteristics, right? So one of the biggest being some amino acids are hydrophilic, some amino acids are hydrophobic, all right? And that's important on protein on the organizational level because it can determine how that protein kind of folds. So the hydrophobic parts are going to kind of be on the inside of a 3D structure. The hydrophilic parts will be on the outside. So if you have a mutation and you exchange, although it's different, one hydrophobic amino acid for another hydrophobic amino acid, then there's a pretty good chance that it might actually fold somewhat normally. If you trade out a hydrophobic for a hydrophilic, that could significantly alter how the protein folds, which could significantly alter its function. Okay. And then last but not least, nonsense, right? And so this has a possible effect on a phenotype, all right? And so in this case, you change from an amino acid to a stop codon. So you stop production. So let's say you have a protein that is 320 amino acids long, right? And the mutation occurs in one of the last codons. And so amino acid number 319 out of 320 is different. Well, in that case, one amino acid at the very end of the sequence might not have a big effect. And so it's still possible that you could have some type of normal function, right? But if you have a sequence that's 320 amino acids long and the mutation occurs at amino acid number 2, so you are short 318 amino acids, well, then you're not going to have function. And we actually refer to situations like that as inactivation or a knockout. So those of you that go into research or maybe have to read research articles, if you've heard of knockout animals, they basically knock out gene expression by purposefully causing a mutation in the genetic sequence that leads to a non-functional protein or no production of that protein. And they can do that by inducing a stop codon. Okay? So this is often lethal. If you heard of mutations that are embryonic lethal, right? So the embryo doesn't grow or the fetus doesn't grow, or maybe it grows a little bit, but then there's like a miscarriage. Sometimes it can be because of nonsense mutations, and the body knows that it can't support life. Okay.
Would a mutation also be considered nonsense if something happened to the start codon?
Ooh, yes. I guess I've never really thought of that scenario. But yeah, I would throw it into there, because if you can't initiate transcription, or if you can't initiate translation, then yes, you'd result in a non-functional knockout. Yeah, that's a good point. See, that's what you're paying the big bucks for, to make me think of new things. Okay, so let's talk about the other major category of spontaneous mutations, and that's going to be nucleotide addition or deletion, right? And in this case, right, I think it was Lauren who asked this question. She talked about a no frame shift, or I asked her a question about a no frame shift in a point mutation. Now, with the addition of a nucleotide or deletion of a nucleotide, we shift those codons, all right? Because if we add something, it changes the pairing of every 3, right, or the grouping of every 3. If we delete something, it changes that grouping of every 3 codons, right? So this is a frame shift. And again, just like before, you have these different outcomes: silent, missense, and nonsense. More often than not, though, if you're causing frame shifts, you're probably going to fall into this missense or nonsense category. You would have to get really, really lucky to cause a frame shift and then have a completely functional protein emerge from the end of that, right? So kind of what does that look like? And this is just a small example. So here again, here's your sequence. So here's your 3 codons, right? In this case, though, we have the addition of a uracil. All right, so we kind of bump it in right between this uracil and this guanine. We add another uracil. And so you can see originally, right, here's your amino acid sequence. We add a nucleotide, and now we have a frame shift. And it is giving you different amino acids. And oops, what did you do? You accidentally induced a stop codon. All right, so you can see frame shift, whether I add or delete, is more than likely going to give you a very different looking amino acid sequence, and then, right, is more than likely going to interfere or potentially could add a stop codon there. And I guess even to Megan's point, I guess you could even add, right, a start codon potentially in the wrong place. All right, we're not going to spend a lot of time on transposons, but what I want you to know is this is basically, I like to call it jumping DNA. And again, kind of like when you guys were talking to me about transformation and the idea that cells had to be competent, in order for transposition to occur, you have to have a transposable element. So this is not just saying any DNA can jump, right? And here you see a circular chromosome, so we're referring to prokaryotic cells. DNA doesn't just randomly jump. You have DNA that essentially is prone to jumping, all right? So you have something called a transposable element. DNA in this other part of this plasmid or chromosome, right, whatever this is representing, is not going to just spontaneously jump, right? It has to be a transposable element. So there are parts of the sequence that make it essentially jumpy or willing to leave. And what we see, right, is that transposable elements can jump to and insert themselves, right? So in this case, you could say, here's a circular chromosome, here's a plasmid maybe, or it could go the opposite direction, right? But they insert themselves into this gene and they interrupt expression or function of this gene. So a good example here is corn. And so you can see these really cool kernels, in my opinion, that are kind of multicolored. So a transposable element has actually been inserted in this case and is interrupting pigmentation in these kind of multicolored kernels, all right? Now, it's possible, and this is the thing we're going to refer to multiple times, that there could be some really vital piece of information that's in this transposable element. We then potentially spread it to this plasmid, and then you guys talked about conjugation, and then you could share that plasmid, right? And so this is a way that genetic information, perhaps things that are pathogenic or bad for us, could be shared between different genetic components of bacterial cells. But it also, right, is a way that just a normal important gene could be interrupted. All right, and we'll revisit that maybe a little bit later one more time, kind of at the end. All right, so we talked about this idea, right? So again, this is focusing on transponase is basically saying you need a special enzyme to do this, right? It's going to operate on that transposable element. And so again, you're not going to just have DNA that jumps. So you're going to need this enzyme present, which is going to be part of the sequence of that transposable element that contains this enzyme that can help cut it out. And often what we see, right, a move between plasmids of chromosomes, non-homologous. Can anyone remember from back in the day, like we said, like homologous, what does that mean between like two chromosomes? If we have a homologous set of chromosomes, right? Does anyone remember? Lauren, do you remember?
They're like the same.
They're like the same, right? And so in that case, if there's a homologous move, right? So like, let's say this contained the gene, and I know it's not as simple as this, but the gene for eye color. And so then it switches, or it enters a new chromosome, right? Or new whatever you want to say, right? A new chromosome. Then it's going to enter at that same area, right? So you're basically replacing a like sequence with a like sequence. It might be a variation, right? It might be a different color, but the gene for eye color is going to replace the gene for eye color. Non-homologous means that it's inserting, in this case, there's no information about gene X in here, but it's going to insert itself and plant itself right in gene X, right? So it's a different kind of set of genetic information that's inserting itself into, okay? And when we talk about horizontal gene transfer, we're going to be referring to a lot of homologous changes in that case. And so I'll kind of point that out again so you can kind of see some differences there between homologous and non-homologous. All right, so let's talk about inducing mutations, right? And one of the ways that we do this is via chemicals, right? So we talked about with my favorite Ninja Turtles there, right? There's some environmental stimulus, right? In interaction with the environment that induces a mutation. And so we have a couple different ways that can happen with chemicals. Now, the important thing to remember with chemical induced mutations is that it's changing the hydrogen bonding characteristics of nucleotides. So remember, between AT, right, you have 2 hydrogen bonds. Between GC, you have 3 hydrogen bonds. So kind of keep that in mind, and you'll see what I mean here in just a second. You have a couple different options: modification of nucleotides, replacement of nucleotides, and then physical displacement of nucleotides. And you'll see what each of these looks like here in just a second. So when we talk about modifications of nucleotides, one of the modifications that we talk about is something called alkylation. Alkylation is simply the addition of carbon chains. So here you have guanine and cytosine, right? Boom, 1, 2, 3 hydrogen bonds. We then add, though, right, a methyl group, which is basically this short 2-carbon chain. We add that to this oxygen. When we add that to oxygen, right, we actually lose the ability to have a hydrogen bond right here. And in fact, we now only have 2 eligible spots for hydrogen bonding, all right? Thymine says, oh, you have 2 hydrogen bonds. That's what I like, right? It normally bonds with adenine. And so now you essentially have a G with a T, okay? That's not good. So now, depending on where this has occurred, right, you are reading through a nucleotide sequence and you see a T where you should have seen a C, and that could totally alter the codon, right, to what you're working with. All right. You also have replacements and you have base analogs. So in this case, we actually have chemicals that resemble nucleotides. So here, right, you would have, right, thymine, which would normally bind with adenine. So here's your thymine or thymidine, right? Call it whatever you will. And here you have bromodioxyuridine. We also just call it BirdU. And if you look at these two molecules, they look very, very similar. The only difference is that you have a bromine here, all right? And because you have a bromine here, you are altering, right, your hydrogen bonds. And now, right, BRDU, which is close to thymidine, can actually bind a G, right, a guanine. And so now you essentially have, right, or you've introduced a different nucleotide, right? We'd want an A there if everything was normal, and now we have a G. And so again, when you start to think about the codons, that can have a very negative effect. And what's really interesting is that this is a really powerful research tool. And so BRDU, what we've done is we've developed antibodies to it in a laboratory setting. And what you can do is you can inject animals with BRDU, and their cells can fuse it for a base, right, for a nucleotide, and they incorporate it into their cells. And when they incorporate it into their cells, you can then, after you've harvested their tissue, right, which is a terminal procedure, in this case for these animals, you can section it and stain it on a microscope using these antibodies, and you can actually use a fluorescent antibody. When you look under a microscope, it actually allows you to identify areas where there is rapid cell division, right? So you could, if you're studying a cancer model, right, you could inject this into them. Because cancer cells divide so quickly, it gets incorporated into them very easily, and then you could look at that tumor under a microscope and it would fluoresce. We actually use BRDU. One of the highest points of turnover in your cells is actually your intestinal epithelial cells. And what you could do is you could basically induce an intestinal injury in animals, and then you could inject them with BRDU, and you could look at their tissue under a microscope. And the more BRDU you saw, that actually was indicative of tissue repair. That meant that the cells were trying to divide more quickly to repair the tissue that had been damaged, right? So it's a very, very powerful research tool. But if you were to continue living, right, which a lot of times laboratory animals are not, because we need to study their tissues, this would not be something that you'd want injected into you, because it would induce mutations. But if you're sacrificing an animal for research, you're not concerned about those mutations because you don't let them go for prolonged periods with these alternate base pairs. Okay. So for physical displacement, this is something called intercalating agents. And what we see here is this is actually very similar to the addition or subtraction of nucleotides. So think about an intercalating agent is if they're, they're taking in every 3 people into your favorite, well, now because of COVID, we'll say into your favorite, let's see. Well, I'll say I love Bed Bath & Beyond, right? So maybe they're taking people into Bed Bath & Beyond in 3 intervals. I love to cook and bake, and the number of cooking and baking options in Bed Bath & Beyond is mind-boggling, right? And so they're taking every 3 people in, and so you have, I'm just looking at my little screen here, you have Rachel, Rachel D, Sarah, and Anna, right, are the first 3 that I see on my screen right now. So they're the first 3 that go in. But then Noelle comes and she inserts herself in between Rachel and Sarah. So now it becomes Rachel, Noelle, and Sarah, and poor Anna is left outside of Bed Bath & Beyond, right? And so Noelle, in this case, is that intercalating agent, right? She comes in, she wedges herself in between Rachel and Sarah, and now we essentially have a frame shift, right? The nucleotides that are allowed into Bed Bath & Beyond, or the codon, has changed, right? It's been altered. And so that's what we kind of see happening with these intercalating agents, right? So it confuses us when we're translating. And here, right, you see, I don't know if you've heard of ethidium bromide. Ethidium bromide actually is an intercalating agent that interacts with DNA. And so you see this little kind of cool little neon pink. These are actually base pairs, right? So these might be gene products of DNA, and we actually shoot ultraviolet light through the DNA gel, and it causes ethidium bromide to fluoresce. And so that's how you can actually look at bands of DNA. Now, because ethidium bromide was shown to cause cancer in rats, it's not used in an undergraduate setting, but we still use plenty of it in graduate work and upper level research. And it's actually really fun. I've heard of multiple stories of older professors that are like in their 60s and 70s and above, still working with ethidium bromide without any type of gloves or sticking their hands in solutions that contain ethidium bromide. And typically their argument is it causes cancer in rats, and they're not rats, and so they don't care. So, I mean, I'm so excited to get into my 70s or 80s and just take that approach to life, right? Sometimes I'm jealous. But I would say that it probably does cause cancer, so we don't use it in undergrad setting. Chloroquine, which is actually an anti-malaria medication, can also be an intercalating agent. So this is one of multiple reasons that if you're going to an area where malaria is prevalent and they give you chloroquine, they'll tell you to start taking it right before, taking it during, and then take it for a little while after, and then get off of it. All right. And then that's it, because although it works really great for malaria, it can also have some negative side effects. Is chloroquine, is that what the kind of the rumor is for COVID-19, right? Is that what they're using? Or some people?
That and hydroxychloroquine.
Okay. Yeah. So, as you've seen, and I haven't really read into it, even though supposedly our president's doing it and taking it, right? Please don't take it without medical supervision, right? This was kind of one of like the first things that came out as a potential, I guess there's a research paper that, showed, or some groups that said, hey, this might be beneficial. And then people jumped with it. And apparently it's relatively easy to get without a prescription if you're buying from other countries. So beware. Right, this is not necessarily a good COVID treatment. It's great for malaria, but there's a reason you don't take it for prolonged periods of time. All right, so inducing mutations, this is not necessarily chemical, but this would be involved, this would be a case of radiation, right? And so you have a couple different options. You have UV dimers, all right? So in this case, UV radiation actually causes covalent bonds to form between thymine dimers, right? And so when you have these bonds that form between thymine dimers, again, that's going to interrupt the replication, the transcription, and the translation process when you kind of pull these nucleotides together. This is, right, a big reason that you want to wear sunscreen when you're out in the sun, right? If you start have this happening to your DNA, right, this can induce mutations. These mutations could eventually lead to cancer, right? X-rays, which it doesn't show on here, but X-rays are actually great at breaking apart this sugar phosphate backbone. And so upon exposure to X-rays, you can have damage to this backbone, and then you can have instability in your nucleic acids, all right? So those of you that have gotten X-rays or have been to the dentist, all right, they typically sit a lead vest on top of you. And this is one of the reasons that they do that. Specifically for females, they're gonna put it kind of across your pelvic region to protect your eggs and your ovaries, right? Because those of you that have had ANP2 probably remember that the eggs that you're born with are the eggs that you got, all right? And so once those are damaged, they're damaged. And so it's really important to do this for females. For males, right, sperm turnover is relatively rapid. And so I'm not saying that it doesn't matter, for males, but it certainly does not matter quite as much, because even if the DNA of sperm is damaged, right, there's the opportunity for turnover there that doesn't exist in females, right? So exposure to UV radiation and X-ray radiation can be damaging to both prokaryotic cells and eukaryotic cells. So how are these things being repaired, right? And they are, right? This is stuff that we encounter. This is stuff that they encounter. So how can we repair it? Well, one of the ways that repair occurs, right, and this occurs in both us and prokaryotic cells, is via something called proofreading. And this is actually going on during replication and happens to be a function of DNA polymerase. So not only can DNA polymerase lay down the new nucleotides, right, as we're replicating a strand of DNA, it also has the ability to slightly back up, right, and read what it's done, right, proofread, right, look at its handiwork. And the way it detects this is kind of cool. And let's see, do I have a, no. Well, I'll show you. Basically what happens is when you have nucleotides that are mispaired, it causes an actual small kind of physical bump in the strand of DNA. And DNA polymerase can actually detect or read that little bump, right? Mispairing causes a change in physical structure. It can detect that change in physical structure, and it can repair its mistake, okay? You also have, right, the ability to look for mismatch that DNA polymerase may have missed, right? DNA polymerase itself, like you when you proofread, is not perfect, right? So when we look at mismatch, here's two complementary sequences, anti-parallel of DNA. All right, boom, I have a mutation now, right? So a DNA polymerase missed this. It made a mistake. There's a T bound to a G. It didn't catch it, even though there might be a physical distortion there. So what mismatch enzymes can come in and do is they can actually come in and they can replace, right, this mismatch with the correct nucleotide. Now, what is maybe a little bit of a dilemma with this situation that you can think of, right? What's the dilemma here? So we have a T bound to a G, so we know that's wrong. So we want to fix that.
Is it that the cell doesn't know which one is the right one?
Perfect, right? That's what we're going to go with. So I show you that, and you can look at that right now, and you can say, Professor Daft, G does not go with T, right? Just like Amanda said. But if I didn't have that T highlighted red, right? I'm telling you that's a mistake, but if I didn't have it highlighted red, how would you know, right? Nucleotides that are bound incorrectly are not highlighted red inside your cell. So how does the cell know? Well, what's really cool is that the longer that DNA is around, it gets methylated, right? And that's fine. This is a normal part of function. If any of you have heard of epigenetics, methylation is actually really important in controlling gene expression. I don't know if you guys cover that in genetics or not, but if you go on to any type of science career or research after this, that probably will come up. So the longer DNA is around, the more it becomes methylated. And so what the body says here is, okay, so this strand, right, is methylated. And so that tells those mismatched enzymes to leave it alone and say, okay, so this is the kind of the old tried and true. It's been around for a while. So we're going to say the mistake then is in this newly synthesized strand, which has not had time to be methylated. And so these enzymes are allowed to interact with it and then replace that base pair. Now, when they replace it too, they remove corresponding nucleotides on either side, right? They can then lay down the correct nucleotide sequence, and we are good to go. Now, the problem would be, right, theoretically, if those mismatch enzymes missed that change and then, or missed that mutation, so then you have an incorrect base pairing. That incorrect base pairing gets, or that strand gets methylated because it's been around for a while, and then you're not able to distinguish, right? So this is not a perfect system. Proofreading, mismatch repair, these special mechanisms we're about to talk about in bacteria, right? It's not perfect, but it is a way that we can catch some mutations, right? So let's actually start to talk about now some repair mechanisms that are unique to prokaryotic cells, right, when we compare them to our cells. So this is a repair of UV damage. So remember I said UV damage causes thymine dimers. That means between all these T's that are highlighted red, all these thymines, they are now actually stuck together with covalent bonds, right? So covalent bonds are holding these T's together, these T's, these T's, these T's, these T's, right? It's not the bond, right, with the anti-parallel strand. It's the thymines that are bound together within the same strand of DNA. Okay? So you guys can't repair these, right? This is why it's important to protect what you have, right? Exposure to the sun, you need to be careful. Bacteria need to be careful too, but, right, they do have a special mechanism, right, that essentially acts like sunglasses on their DNA, right, on these thymine dimers that have formed. Right? And one mechanism is called light repair. So this is only in prokaryotic cells. Light repair is using actual UV radiation to help power enzymes that can come in, and they can actually break the covalent bonds between these thymine dimers. Now, right, that means that the original nucleotides that you had before stay there, right? We simply come in and we separate them, right? Kind of break those covalent bonds that hold them together, and then you're good to go. We also see in some, and I see that Snape because he was one of the Dark Lords, right? We also see dark repair. And dark repair is also only in prokaryotic cells. And in this case, dark repair is removing nucleotides that are on either side of those thymine dimers, laying down a new set. And when you lay down a new set, you lay down thymines that are no longer bound together, and you now have your repair, right? So light and dark repair, and you'll see at the end of this, there's a little table that actually shows some of, or kind of lines these different types of repair up for you guys. The last type of repair that we're going to talk about that's unique to prokaryotic cells, and this is one of my favorite, is called SOS, right? So if you've heard of SOS, right, it's a distress signal. So it used to stand for Save Our Ship, right? So ships that were sinking would send out an SOS request, and that would hopefully signal to somebody that they needed help. And this is the same idea in bacteria. So SOS is induced, or SOS repairs induced in prokaryotic cells when there's severe damage, right? So you have nucleotide additions, deletions, you have perhaps frame shifts, you have, you can see here, mismatch, you have thymine dimers, right? It is just a mess, right? There is severe damage. So presumably there's been some type of crazy environmental stress that's induced these changes. And so you have SOS DNA polymerase that comes in, and it just starts laying down nucleotides to kind of try to fix things or fill the gaps. It doesn't proofread. It doesn't care, right? Think about it. If you're, we'll use the ship analogy. If you're in a ship or if you're in a submarine and you spring a leak, you care what you're shoving into that hole, right, to plug it or block water from coming in? No, you're just grabbing anything you have in front of you, shoving it into that hole, and hoping, right, that it keeps that water out. And that's what SOS polymerase is doing. It doesn't care about mutations, right? It is simply saying, in order to survive, I need to have nucleotides present in this spot. And what's going to inevitably happen is the bacteria may survive, but there's going to be a lot of mutations that occur in it. Would you rather be alive and mutated, or would you rather be dead, right? And so bacteria take the approach, or prokaryotic cells take the approach, right, that we're going to plug all these holes, and we're going to deal and live with the consequences of the mutations that we may have, okay? And so why would this be bad in us? What would happen to us if we had the ability to have SOS DNA polymerase and SOS repair? What are some of the negative consequences that we might experience? Right, don't overthink it. What's one of the first things that comes to the top of your head?
Cancer.
Cancer, right? I mean, that's this would be, there'd be a lot of mutations, and a lot of mutations in your cells are not good, and mutations, right, are what cause cancer, right? Those are normal cells misbehaving, and this would just increase the likelihood that those cells would continue to misbehave, right? So it works for prokaryotic cells. It would not work well for us. And so, you know, in some cases, I would consider this a blessing that we don't have a repair mechanism that works quite like this. Right. So here's talking about, you know, how mutations can be induced. And then I think somewhere, I'm sorry, there should, I don't know if I have it in here. Oh yeah, and here, okay, so how mutations are induced and how they're repaired right here. I, for some reason, thought they're in separate charts, but you can see different repair mechanisms: proofreading, mismatch repair, right? Here's light repair, dark repair, SOS repair. So look over those quickly. So we're going to talk about here in the next few minutes, and then we'll take a small break and we'll get started on lab stuff. Is, you know, one of the things that should be interested in is how do we identify mutants when we're talking about bacteria or prokaryotic cells, and how do we identify what causes mutations in these cells. And so these are the two different things we're going to talk about. Now, one thing that I want you to kind of pay attention to is the different type of tests that we use to detect mutations are different than the tests that we use to figure out if something can cause a mutation. So please don't get those mixed up. I'll try to point that out. So when we're talking about mutation selection, sometimes there's kind of two basic groups that we categorize things as, or bacteria as. So if you heard of auxotrophic or auxotrophs, right? These are basically our mutants, right? They require some type of growth factor, some type of addition to their regular media in order to grow. So a lot of times, an example of an auxotroph or a mutation is they no longer have the ability to produce an amino acid. And so you have to add that amino acid into the media in order to get them to grow. So you'll see what that looks like in just a second. Prototrophic cells, these are wild type cells. They don't require the addition of a growth factor. All right. And this is primarily going to come into play in something called indirect selection. Okay. Before we talk about indirect selection, though, and auxotrophs and prototrophs, what I want to talk about is direct selection. And direct selection, the best way to look at it is these mutants can grow somewhere where their parents can't. And so a great example of looking at bacteria via direct selection is looking at antibiotic resistance. And so here you have a liquid culture that contains two types of cells: a streptomycin resistant mutant, which is an antibiotic, and a streptomycin sensitive cell, right? So that means if streptomycin was present, these sensitive cells would die. And so what you can do is you can take two plates. You always want to control. So here's a plate that contains streptomycin in the media. So streptomycin's in this plate. Here's a plate that does not contain streptomycin. You take a sample of bacteria from the tube, you put it on each plate. All the bacteria should grow here, streptomycin resistant or streptomycin sensitive, because there's no antibiotic in the plate. So here you can't distinguish between the two colonies. Here, however, right, if streptomycin's in the plate, the colonies that grow, that means they're resistant to streptomycin. That means they have the mutations, right? This is direct selection, right? Indirect selection. So here's where we really start to talk about those auxotrophs and prototrophs. So here you have a plate. This is a nutrient auger. Those of you that have looked at your packet briefly for lab, TSA is a type of nutrient auger. That basically means that all the nutrients that bacteria could want, whether they're a mutant or not a mutant, are there, and so everything can grow. And so what you can do in this situation is you can take a piece of sterile velvet, you press it onto the colonies you have growing on this plate, and it's basically like making a stamp or a copy, right? Those colonies get stuck, or pieces of them get stuck on this plate. You can then take this stamp and press it onto two different plates. You can press it onto the same type of plate that you had before, this nutrient auger where everything can grow. You can then press it onto something called a glucose salt plate, also called a minimal media plate. All right. So you press it on. Here, right, you have your nutrient auger, so everything grows. You can't distinguish auxotrophs from prototrophs. You can't tell me, hey, this bacteria has the inability to make this macromolecule, or this bacteria has the ability to make this macromolecule, because they all look the same because all the nutrients are available to this plate. However, what we see here on this glucose salt plate, or this minimal media, is that when you press the same stamp on and you compare the two plates, you see here it kind of highlights this small section. Something's missing, right? It's not growing there. And that means that this is the auxotroph. Because this minimal media plate doesn't have all the nutrients that are needed by mutants, it can't grow here. So it's indirect selection because you have to actually compare it back to the plate where everything grew in order to identify where it is. So you're actually pulling it off of the plate where everything grew because it can't grow on this plate, right? So it's indirect. Compared to direct, right? The plate where everything grew is not useful to you. That doesn't distinguish anything for you, all right? So indirect selection. Got it? Toomey, you got that? All right, cool. We'll go with it, right? Please look over direct selection. Please look over indirect selection. And please don't get it confused with something we're about to go over called the Ames test. Now, if you go back here and you look, right, you're like, man, that's a lot of colonies to look through and compare back to just to find your mutant. So what's a way that you can actually limit the number of prototrophs, right, the number of wild types that grow, to make it easier to identify your auxotrophs or those mutants? Well, one of the ways you can do that is something called indirect penicillin enrichment. And so in this case, let's review really quickly. What do wild type cells do in that minimal media? What are they capable of doing? Okay, you're driving for a cup of coffee. Got it. All right, love you girls. All right. So what do cells do? What do wild type cells, right, our normal cells do in minimal media? What can they do? What are they good at doing? What did you say, Rachel D?
They can grow.
They can grow, right? They can divide. They're not mutants. That minimal media, that glucose and that salt, that's enough for them to do whatever they want. I always forget that I need to like ask yourself this. That's what should pop up first. But at least question 1 pops up before question 2. So that's something, right? What do mutant cells not do in minimal media? Glory? Did you say something?
I said, because
Okay, perfect. Someone help Glory out a little bit. What do mutant cells not do in minimal media?
They don't grow.
They don't grow, right? So if you have a mutant and a non-mutant and you put them in minimal media, the non-mutant, the wild type, is going to divide and grow easily. That mutant, something's missing. And so it's just going to kind of sit there for a while, right? It's almost like that mutant would be stuck in that stationary phase in the growth curve. All right. Allie, can you remind me quickly, how does penicillin work? What needs to be happening for penicillin to work?
Does it attack the cell walls?
Attacks the cell walls as what? As they're being...
Oh, like as they're dividing?
As they're being assembled, right? As those bacterial cells are dividing. So penicillin, right, only works if your cell's dividing. And so we actually use this to our advantage for indirect selection. So what you see here is that you take your 2 bacteria and you put them into a glucose salt broth, right? So this is just like the plate, that minimal media plate. This is a minimal media broth. And so what you do, right, is you then take a sample of this, right, and you can put it directly on a plate like you did before. And you can see here that everything will grow on this nutrient plate. Unfortunately, right, bacteria in real life, if they're mutant or non-mutant, aren't different colors. And so that doesn't really help you. So what you would do here is you then go through that process of indirect selection, and you'd have to kind of tease through all these colonies. A kind of little bit of a trick is you can actually add penicillin to this tube. And when you add penicillin to this tube, it's going to kill the wild type because they're able to divide. And it's going to, for the most part, leave the mutants alone because they're not able to divide. Since they're not dividing, they're not building new cell walls, and penicillin doesn't really do anything to them, right? Before you add it, though, to this nutrient-rich plate, you actually add an enzyme penicillinase to inhibit penicillin, right? Because if you didn't add penicillinase, there'd still be penicillin in the solution that you put on the plate. They would start dividing because they love the nutrients in this plate, and they would all die, right? So that wouldn't be good. So you have to add penicillinase to neutralize the penicillin. You put them on the plate, and you now have a lot fewer wild type cells present on this plate than if you didn't add penicillin. And so it's easier, right? Because there's fewer colonies, you have to do less work when you're going through indirect selection. Now, just because you did penicillin enrichment does not mean that you can just look at this plate and figure out what's what. Because again, it shows you here that they're different colors, but they're not. And so with this plate, you'd go through the whole process of indirect selection. You're just working with fewer colonies, and so it's easier to identify your mutants. Does that make sense? This doesn't replace indirect selection. It just makes indirect selection easier when you go through that process. Capiche? Capiche. All right, cool. Please look over this. It may seem a little confusing now, but I want you to be able to put this together. So if you struggle, if you're confused, please shoot me an email. Let me know. We can Zoom. We can do it just through email, but I want to make sure this is kind of clear in your minds. Now, let me see here. I want to see something. There we go. Whew. All right. What I'm going to do is we're going to talk about the Ames test. That'll be the last thing we talk about. Professor Perry is going to talk about fungi tomorrow, but she'll do that like in the first half. So we'll let her talk, and then we'll kind of finish this up because there's a few slides here. I just want to go over horizontal gene transfer again with you guys. So we'll do that tomorrow after the fungi lecture, and then we'll also talk about maybe some algae a little bit tomorrow. So I'm going to finish up here with the Ames test, and then we'll finish the rest of this after the fungi lecture tomorrow. Okay. So what we focused on with direct selection and indirect selection is how do we identify a mutant, a bacteria colony that has mutated. The Ames test is now looking at, can a chemical be the cause of a mutation, right? And a chemical that causes a mutation, we call that a mutagen. So indirect selection, direct selection is actually looking for a mutant. It's looking for a mutated colony. The Ames test is looking for a mutagen, something that causes a mutation. So typically when we're looking for something that might cause a mutation, right, we're working with a type of salmonella that does not have the ability to make histidine, right, which is an amino acid, not histamine, histidine. And so what we do, right, is we utilize these glucose salt agar plates, right, these minimal media plates that lack histidine. They're going to be an important part of this experiment. What we also can include in this experiment, and I'll show you on the next diagram where this occurs, is animal liver extract. One of the things that's cool about the liver is a lot of chemicals that are put into your body, including medications, will actually go through the liver. And the liver, a lot of times, modifies these medications or these chemicals. And sometimes when it modifies medications or chemicals that were maybe not toxic before, after they pass through the liver, they could actually become toxic. And so when you are testing the ability of something to be a mutagen, sometimes it's nice to actually expose this mutagen, or this thing that you think is a suspected mutagen, to enzymes that typically would be seen in the liver to see if it actually makes it toxic. So this is the Ames test. Please note, this is important. You have to have a control plate and a test plate. So what you do in your control plate, again, remember this is that minimal media. You add your bacteria, right, that can't grow at all. You add them to your plate. You add a control liquid that does not contain your mutagen, and then you incubate. And what you see here are actually the presence of 2 colonies. And what this is showing is this is spontaneous mutation. This is actually an example of a reversion, right? So these bacteria that you put on the plate couldn't make their own histidine before. However, you see that a couple of them spontaneously mutate and can now make their own histidine, so they're able to grow, right? But that's going to be very, very few colonies. Keep this in mind, right? We're coming back to this control plate. Your test plate, all right, you do the same thing. You put bacteria on it that can't make its own histidine. The plate lacks histidine, so these bacteria shouldn't grow. You then add a liquid with your expected mutagen. If you have animal liver extract present, this is where you also add it so those things can interact. If this liquid on its own, or if this liquid with the addition of an animal liver extract, right, and typically we use rat liver extract, is a mutagen, what you will actually see is a lot of colonies growing, right? So now, right, these colonies have mutated or reverted back to, because of the mutagen, to a colony that can grow in the absence of histidine in the plate. All right. If the chemical is not a mutagen, well, then you'll only see a couple colonies, and these colonies, again, are here because of a spontaneous mutation that occurred in the absence of the mutagen. The reason that you need to have this control plate present is because if you didn't do a control plate without the mutagen, you'd look at this and you could maybe tell me, well, they grew because a mutation was induced. It didn't work in a lot of them, but it did work in some of them. However, if you have that control plate where you didn't have the mutagen and it was just spontaneous mutation, you compare these two back. Hey, this plate looks really similar to the control plate. So that means that this was just spontaneous. It didn't have anything to do with the mutagen. So this chemical is not a mutagen. All right. So that's why it's really important to have the control plate so that you can tell, hey, this was just spontaneous mutation. It didn't happen because of the mutagen. Well, this clearly happened because of the mutagen. All right. Cool. So what we'll talk about briefly tomorrow after Professor Perry talks to us a little bit about fungi are some spontaneous mutations, and then maybe a little bit of algae, or we'll talk about something, right? Dealer's choice. I'll decide what we kind of go over then. All right. So what I'm going to do is I'm going to stop this lovely